Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa. In today's video, we are going to continue on with the nervous system, looking specifically at neuron histology. Hopefully you've had a chance to take a look at last week's video where I gave an introduction to the nervous system. In this video, we'll be taking a look at 6 different types of neuroglial cells, where they belong, whether it be the central nervous system or the peripheral nervous system. We'll be taking a closer look at myelination and how it happens in both of those systems. And finally, we'll be talking about regeneration of neurons in the peripheral nervous system. If we take a look at neuron histology, taking a closer look at nervous tissue, you'll notice that nervous tissue is composed of 2 different types of cells: neurons, which I talked about in my last video, and neuroglia. There are about 1 trillion neurons in the nervous system, and neuroglia outnumber the neurons by as much as 50 to 1. Neuroglia are also referred to as glial cells. So in the nervous system, what you'll see within that nervous tissue again is neurons and neuroglia. Let's talk more about this neuroglia or glial cells. They support and protect the neurons that we talked about last time. They bind neurons together and form the framework for nervous tissue. In the fetus, they're responsible for guiding migration of neurons to their destination. And if a mature neuron is not in synaptic contact with another neuron, it is then covered by glial cells. This prevents the neurons from touching each other and gives precision to conduction pathways. So neuroglia are very important support cells that we find within the nervous tissue. There are actually 6 different types of neuroglial cells, some that are found in the central nervous system and some that are found in the peripheral nervous system. We're going to talk about each type, what they do, and where they're found. Out of the 6 different types of neuroglia cells, 4 of them are found in the central nervous system. We are going to talk about these 4 central nervous system neuroglial cells first. The first one we're going to talk about are astrocytes. Astrocytes are actually the most abundant glial cell in the central nervous system. They have very diverse functions, which include they form a supportive framework of nervous tissue. They have extensions, which are referred to as perivascular feet. These extensions connect blood capillaries that stimulate them to form a tight seal called the blood-brain barrier. The blood-brain barrier is really important because it doesn't allow everything to travel from the body into the brain area. It forms an extra barrier there so that only certain small particles can come in from the body into the brain. They are capable of converting blood glucose to lactate. This is also really important. Obviously, the brain needs sugar in order to function, and that is in the form of this lactate. So these astrocytes are responsible for converting blood glucose to lactate, and then they supply this to the neurons for nourishment. They also contain what are called nerve growth factors. These are secreted by the astrocytes, and they promote neuron growth and synapse formation. They communicate electrically with neurons, and they may also influence synaptic signaling. They also regulate chemical composition of the tissue fluid by absorbing excess neurotransmitters and ions. As the neurons do their work and release neurotransmitters and ions come in and out of the neurons, there could be an excess of this in the extracellular fluid. So the astrocytes are actually responsible for helping to regulate that chemical composition of the extracellular fluid around the neurons to make sure that the neurons can always work at their best. And finally, astrocytosis or sclerosis is when a neuron is damaged. The astrocytes are going to form hardened scar tissue and fill the space that was formerly occupied by that neuron. So as you can see, astrocytes are not only the most abundant glial cell that we find in the central nervous system, but they have all these diverse functions that are really important for the action of the neurons. So those astrocytes are really acting like a great support cell, helping those neurons to work to the best of their capability, providing them with nourishment, making sure that extracellular fluid stays maintained. And so they're really acting as a great support cell within the central nervous system. If you've forgotten already, remember that the central nervous system includes the brain and the spinal cord. The next type of neuroglial cell that we find within the central nervous system are oligodendrocytes. Oligodendrocytes form the myelin sheaths that we see in the central nervous system. They have arm-like processes that wrap around a nerve fiber. This forms an insulating layer that speeds up signal conduction. We'll talk more later about myelination. Ependymal cells are also found within the central nervous system. These cells are responsible for lining the internal cavities of the brain. They are actually a cuboidal epithelium with cilia on its apical surface. Remember, the apical surface is the surface that is going to be towards the cavity, so it has cilia on that side, those little hair-like structures. And these ependymal cells are important because they're responsible for secreting and circulating cerebral spinal fluid. And so we see them within these internal cavities of the brain, within these ventricles. They're going to be making cerebral spinal fluid, or CSF, and they're going to be secreting it, circulating it, etc., throughout the central nervous system. The cerebral spinal fluid is really important within the central nervous system. We find it within the brain and with the spinal cord, and it helps to keep everything well maintained, clear nutrients into those different areas, make sure ions are in those different areas, etc. Finally, the fourth type of neuroglial cell that we'll find within the central nervous system is microglia. Microglia are small wandering macrophages that are formed by white blood cells, which are called monocytes. If you remember what monocytes and macrophages are, monocytes can become macrophages. So these specialized macrophages that we find in the central nervous system are referred to as microglia. Remember, macrophages eat things. Those cells can undergo phagocytosis, which means cell eating, and so they're basically cleanup cells, picking up any debris. So it is thought that they perform a complete checkup on the brain tissue several times a day. These cells are also unique because they're wandering, which means they can move around. So they move around, they wander in search of cellular debris, they eat that up, they maintain this and make sure that it's really clean. So these four cells that we find within the central nervous system, again, astrocytes, oligodendrocytes, ependymal cells, and microglia, are really important to maintaining the environment within the central nervous system, within the brain and spinal cord, so that the neurons can function properly and send their signals as they should be able to. In the peripheral nervous system, we see two types of cells, Schwann cells and satellite cells. Again, these are neuroglial cells, and we find them within the peripheral nervous system. If you don't remember what the peripheral nervous system is, the peripheral nervous system includes the spinal nerves and the cranial nerves. So the nerves that extend from the brain and the spinal cord would then be the peripheral nervous system. So two different types of neuroglia we see here again are Schwann cells and satellite cells. Schwann cells are similar to oligodendrocytes. Schwann cells are going to produce the myelin sheath that we see within the peripheral nervous system. Remember, oligodendrocytes produce the myelin sheath in the central nervous system. They do it a little bit differently. The Schwann cells are going to envelope the nerve fibers within the peripheral nervous system. The cell itself is going to wrap around rather than arm-like processes like we saw in the central nervous system. And this is going to produce that myelin sheath and also help to assist in the regeneration of damaged fibers. We'll talk more again about myelination and go into a little bit more detail of the difference between the Schwann cells and the oligodendrocytes in just a minute. The other type of cell, neuroglial cell, that we see in the peripheral nervous system are satellite cells. These cells surround the neurosomas in the ganglia of the peripheral nervous system. Remember, in the peripheral nervous system, we have what are called ganglia. And ganglia is where all the cell bodies of the neurons hang out together in one outswelling, and then their axons would extend out of there. So because all the cell bodies hang out together in this ganglia, creates this swelling. So there's swellings within the peripheral nervous system of this, and the satellite cells actually hang out there. They surround those cell bodies or neurosomas in there, and they provide electrical insulation around the soma, and they also are responsible for regulating the chemical environment of the neurons within that area, okay? And that means, again, maintaining that extracellular fluid, making sure that everything is good and ready for the neurons to be able to conduct their signals. Neurons are very specialized cells. They're capable of this conductivity. They can send signals to one another, just like muscle cells can. And these are very special qualities that these cells have that the other cells of our body don't have. And in order for them to be able to receive these signals and send these signals properly, the extracellular fluid has to be maintained around these cells. And that is what the satellite cells do within the peripheral nervous system. In the central nervous system, we have the astrocytes that are doing that. I mentioned we would talk a little bit more about myelin. The myelin sheath is an insulating layer around a nerve fiber. In the central nervous system, it is formed by oligodendrocytes, while in the peripheral nervous system, it is formed by Schwann cells. It consists of the plasma membrane of glial cells, and the plasma membrane is mostly lipid, those phospholipids that make up that bilayer of the plasma membrane. So it's about 80% lipid and 20% protein. And myelination, that term, is given to indicate that that is the production of the myelin sheath. So we're going to have a myelin sheath on these axons. How is it formed? It's formed through myelination. It actually begins the 14th week of fetal development and occurs very rapidly during infancy. This whole process is completed in late adolescence. And it's really important you hear about dietary fat, and it's really important in this process so that the nervous system can develop. In the peripheral nervous system, the Schwann cell itself spirals repeatedly around a single nerve fiber, and it lays down as many as 100 layers of its own membrane. There's no cytoplasm between the membranes. And so if you see an axon in the peripheral nervous system, what you'll notice is that this one cell itself wraps again and again and again around a certain area. So it's cell and cell and cell where it's doing this wrapping. In the central nervous system, oligodendrocytes are going to reach out to myelate several nerve fibers in its immediate vicinity. And so it extends these arm-like processes, and those arm-like processes is what's going to do the wrapping around the cell. So in the central nervous system, you'll see an oligodendrocyte with its cell body, almost imagine like an octopus, extending many arms out, and those arms are actually doing the wrapping. So one cell in the central nervous system, one oligodendrocyte can wrap in many areas along the axon. Whereas in the peripheral nervous system, one Schwann cell can only wrap one area of one axon. So the way that they both do it is very different from one another. What happens is, no matter where this happens, whether it be the central nervous system or the peripheral nervous system, the myelin sheath is segmented. And because it is segmented, it creates these gaps between segments that are referred to as nodes of Ranvier. And the internodes is where you will actually see the myelin. So there'll be a myelin coverage, there'll be a little gap, myelin coverage, a little gap. And so where there's a myelin coverage, those are referred to as internodes. Those gaps are referred to as nodes of Ranvier. The trigger zone is going to be where the axon hillock and the initial segment are. So where the axon comes out from the cell body, that's where the trigger region, the trigger zone is going to be, where an action potential would start. So we're setting up for the idea that if an axon is myelinated, okay, it is going to conduct the signal much quicker because the signal is going to come from the dendrites through the cell body down the axon. If an axon is myelinated, then the signal is going to conduct much faster because it's almost as if it jumps from node of Ranvier to node of Ranvier to node of Ranvier. So it occurs much quicker. If the cell is unmyelinated, then it's going to take longer for the signal to pass through the axon, okay? So myelinated, signal happens much faster. Unmyelinated, signal takes a little bit longer to pass through the axon itself. So the conduction speed of nerve fibers is going to depend on a couple of different things. One, the diameter of the fiber, and two, the presence or absence of myelin, which I just spoke about. So if the fibers are larger, then they're going to have more surface area and they're going to conduct signals more rapidly. Myelin also is going to increase the speed of the signal conduction. So for example, if you have a small unmyelinated fiber, it's going to conduct about 0.5 to 2 meters per second. But if you have a small myelinated fiber, so the only difference, they're both small, the only difference is that this one is now myelinated, it would be able to conduct at about 3 to 15 meters per second. If you have a large myelinated fiber, those can conduct up to 120 meters per second. And so why would we want the difference between a small myelinated, small unmyelinated, these large diameters? Why do we want the difference? Now here's what's important. The slow signals are going to supply the stomach and dilate the pupil, do all these types of things. Those are examples where speed is less of an issue. Maybe it doesn't need to happen too fast. The fast signals, though, are going to supply the skeletal muscles and transport sensory signals for vision and balance. So when we see something, we want to be able to process that quickly, right? Or we don't want to bump into that. So that's important that that happens quickly. If we touch a hot stove, that sensory signal, we want that to be able to send the motor signal very quickly for us to move that hand away from that stove. So there are things that we want to travel very, very quickly to happen at a quick pace. And there's other things like digestion processes, etc., that doesn't necessarily have to happen that quickly. So depending on where these neurons are innervating is going to depend on what type of neuron, small, large, myelinated, unmyelinated, to depend on how quickly that signal is going to be sent. Let's talk a little bit about the regeneration of peripheral nerves. Now, the brain and the spinal cord, the central nervous system, the peripheral nervous system, these are all areas that are really being studied a lot. And we don't know everything about these areas yet. But what we do know is that if there's damage to peripheral nerves in the peripheral nervous system, that some regeneration can occur depending on how badly damaged that nerve was. As of right now, damaged nerve fibers in the central nervous system cannot occur at all. And so there is some differences between this. In the peripheral nervous system, damage to a peripheral nerve fiber can occur if the soma is intact. So if the cell body is still intact, if there is at least some neurolemma that remains, so there's some plasma membrane that is still there, then what's going to happen is that there's going to be a regeneration tube that's formed, and this is formed by those Schwann cells that myelinate within the peripheral nervous system. The basal lamina, which is the bottom layer of the cells, and then the plasma membrane of the axon itself, so the neurolemma near the injury. The regeneration tube is going to guide the growing sprout back to the original target cell and reestablish a synaptic contact. So this is really going to depend on how far away this is, how long this is going to take, etc. And once this happens, if this is able to happen, if the neuron isn't damaged that badly, then once this reestablishes, the nucleus can return back to its normal shape. So again, this is something that can happen within the peripheral nervous system. It does take time for this to regenerate, but as of right now, it is not something that can occur within the central nervous system. Thank you so much for watching my video. I hope that it's helping you as you study for your classes. Maybe you're just wanting to learn some information. I hope that through my videos that your knowledge can grow. One of my biggest goals is to be able to take these difficult topics and make them easy to understand. If you have any questions about this video, please make sure that you write your comments below and I'll answer them as soon as I can. If you ever have an idea for another video that you'd like to see, some kind of topic that you might not be able to find, drop that down below too. If you like the content of my channel and my videos, please make sure that you subscribe and click on the notifications so that you never miss out on a new video. Thank you so much for being here, for supporting me, and I hope to continue to bring these videos to you and just to be able to help you better understand these really hard to understand concepts. Thank you.